Good afternoon, everyone. So I'm Ibrahim Bitar. Thanks for the introduction. So today we're going to talk about the ups and downs of microbial genomics in the era of long reads. So bear with me for 20 minutes. It's going to be interesting, hopefully. So we're going to talk about, half of my time would be talking about the struggle of dealing with short reads in microbial genomics, and then how we shifted going from Illumina to PacBio, and now I can say that we're actually ditching short reads for good after the introduction of Vega. So if I'm going to talk a little bit about the microbial genomics, it's important to describe a little bit about the chromosomes and the structure and what we search for in epidemiology when dealing with microbial genomics. So normally, we deal with antibiotic-resistant genes. These genes actually are carried by what we call insertion sequence or repetitive mobile genetic elements. And these mobile genetic elements, normally, as you can see in the red color, will actually carry these antibiotic-resistant genes and insert it in what we call a transposon, another mobile genetic element, which also can contain multiple antibiotic-resistant genes that can be also part of a mosaic structure of different transposons and integrons and so on. So these mobile genetic elements are made up of integrons, plasmids, transposons, prophages, and you can name it. And they share something in common with all, is that they have a really high sequence similarity, more than 80% in sometimes. And this is very important if we want to talk about short reads. And normally these kind of structures or mosaic MDR regions, multiple drug-resistant regions, go a little bit of modification along the way. So you can lose one antibiotic-resistant genes. You can acquire more antibiotic-resistant genes. It can have also some inversions. And then we're going to talk about what we call plasmid, and this is the most difficult mobile route of dissemination of antibiotic-resistant genes. So I'm not going to go in details with plasmids discussion, just to tell you the, let's say, the definition. So it's a circular DNA structure that you can find within the bacteria. It has its own replication machinery, so it replicates without depending on the chromosome. And normally it's found in a kind of a higher copy number within the bacterial cell. And this kind of the armor of the bacteria, which normally have many antibiotic-resistant genes and many virulence genes, and it can be shared between different bacteria. It doesn't have to be the same species, so an Escherichia coli can actually give it to Klebsiella pneumonia, Klebsiella pneumonia can give it to Acinetobacter, and this is actually the main problem as a microbiologist we face in how to treat or stop this dissemination. So working with Illumina, you think it's really easy. So you have as much coverage as you can. And as you can see, the assembler will look if you have some kind of mis-sequenced base, as you can see right over here. The assembler will correct it and make it an A. But if you have shorter or less coverage within the bacteria, you will have a bit of problematic. So if you have enough coverage, everything is solved. Actually, it's not. So the dilemma with bacterial genomics is that because of this mobile genetic elements, insertion sequences and transposon, these normally are found in a high number in different places in the chromosome and the plasmid. So it's a repetitive element, as we said. So what will happen is, as your assembler is assembling your reads, and then it reaches this insertion sequence, for example, one gene, that is found in two places in your chromosome. So the assembler doesn't know how to go from there. Is it the way up or the bottom? And since he cannot know what to do, it will break your contig into two different contigs. And then it continues. But of course, insertion sequences are found in many numbers, so you will find another insertion sequence or a repetitive element that is found in multiple places in your genome, and then your contig will break again. And so on and so on. And then you end up, for example, if we're going to give a general number of contigs per bacterial genome, you end up with 300 contigs per assembly. So normally what people do is what we call in silico scaffolding, or for example, they don't do de novo assembly and they just map their reads against the reference genome. Actually, I don't like that, but I will tell you in a while why. So, in our kind of antibiotic-resistant microbial genomics, what we do is Normally, we upload our assembled genome into some online databases that can actually tell you where is the position of antibiotic-resistant gene, such as ResFinder. And if it's found in a plasmid, the plasmid has a replicon gene. Also, when you upload it to a plasmid finder, it will tell you where is this replicon of the plasmid. So, for example, we have here the MCR1, and this is actually the gene when Obama was the president and sent out a call for action because of superbugs. It was because of this gene. So it's a mobile colistin resistance. And you can find in our assembly, it was found in node 49 or in contig number 49. And then, because of, we know that normally you can find the MCR on a plasmid called IncX4, you will try in silico to try to connect this contig to this contig. And so you can see the overlaps between the different contigs until you can connect it. But that is, for me, at least, is wrong. So a study done by us in 2017, where we have 4 Escherichia coli plasmids. We were studying the metallobeta-lactamase, BLA-IMP. And we found out in Czech Republic, we found these isolates carrying the same plasmid type, which is an IncAC, and they have the same antibiotic-resistant genes. However, you can see a huge variation in the size of the plasmid, and this is only the MDR region, so it's not the whole plasmid. So if you say that, for example, we found this plasmid, and we recognized it for, 3 years ago, and if you map your reads against this one or this one, you will lose a lot of your actual structure of the plasmid. So this is why assembling across a reference is not always a good idea. Another example that we actually did using our SQL, so we found some, what we call an antibiotic-resistant island, so different islands on a plasmid in the database, but actually we couldn't find the plasmid in our isolate. So we found out that these 2 antibiotic-resistant islands were actually inserted within the chromosome of the sample. Another also examples that we gave, also we did this via the SQL one. So normally the BLA-OXA 181 is found in what we call IncX3 plasmid, but you can see from these 2 samples, it was not on the same plasmid that you found 90% of the times in the literature. It was in a smaller plasmid, and here you can find a fusion of 2 plasmids as well, carrying the same gene. And of course, Klebsiella normally have 8 or 7 or 9 plasmids within the same isolate, and these plasmids are really kind of big, so 100 kb and above. So it really takes a lot of time when you're dealing with short-read sequencing to actually close these plasmids. I'm giving this so much examples just to say that you could say, for example, I would face this problem once in three months. That's not the situation. You could face this every week. So another misassembled chromosome as well. So this was done through Illumina as well. We found out a bit of different coverage for the BLAGS5. So we did it on long reads, and we found out that this gene was actually duplicated in our sample. And finally, there's another problem that not everybody talks about, that not all plasmids are typable. So if you upload your assembly on the plasmid finder, there are some plasmids that are untypable, which in this case carry different integrons carrying the same antibiotic-resistant genes in 2 different species. So what I used to do, what I did is I did what we call transformation. So I extract the plasmid of interest that has this kind of the antibiotic-resistant gene that I'm interested in. And then I will do transformation and insert it in a recipient cell. Normally, it's in a topo 10 Escherichia coli. These recipient cells are a kit that you can buy from different vendors, and the genome is closed and completed. So I would do Illumina sequencing, and then I map the reads against the reference, taking out all the chromosome out, and then you're left with your plasmidic reads. Of course, there are some parts that are found in both the chromosome and the plasmid that you will lose in this thing. And also another thing that you can do is try to do assembly using 2 different pipelines or assemblers in order to see if one assembler will actually produce some contigs that is a little bit longer, and you can close the gap. Another thing you can do is also use, for example, bandage to kind of visually see which contig see which contig. Normally, if you have a transformation, you'll end up with something like this. But if you go with the native bacteria, then you'll end up with a hairball that you cannot see. And eventually, this is why I wanted to drop the Illumina, is because of this procedure, which takes a lot of time and it doesn't always work. So it's called S1 nuclease pulse field gel electrophoresis. So you extract your plasmids, make plugs, and then you digest it with S1 nuclease, which will actually linearize your plasmid. You do pulse field gel electrophoresis, and then you transfer your DNA with a positively charged membrane, and then hybridize it by a double-labeled probes for your specific gene. For example, here we found out that the KPC was carried by a plasmid that is almost 100 kb long. So now you have the approximate length of your plasmid. You have the transformation, so let's say 50 contigs more or less. And then you can do the final step when you're dealing with Illumina, which is the PCR gap closing technique. So for every possible connection, you design primers, do PCR, and the positive one you sequence, fill in the gap, and continue with the same thing until at the end you design the primers that will circularize your plasmid, and you end up with a closed plasmid. Some of the plasmid took me 3 months to close. Some of them never closed. And this is an example, for example, for a 40 kb length MDR region within one of the bacteria. These are the PCRs that actually worked. We designed more than 60 primers, and this is the one that we managed to close only the MDR region. If you see, the plasmid was 240 K, so we didn't close all the plasmid. And then long reads came in, of course, PacBio and nanopore. You all know the technology behind both of them. So, for example, one of the plasmids, which is 240 kb, instead of having 18 contigs, it was one circular contig. But again, you can also see from the blue coverage there that there are some parts that were all actually missing from the Illumina. And then you end up with a one contig for the chromosome, which is instead of 330 contigs. We actually introduced Minayan, and it stayed only for 2 weeks, and then I ditched Minayan. I'm not going to talk about the reasons because I'm not supposed to curse here, so maybe we'll do it later. So, but again, you know that you can do hybrid assembly using unicycler, for example, and they are still developing and trying to develop different pipelines for assembly with nanopore. So you can do assembly using the long reads and then do kind of Illumina polishing, and then to have a bit more accurate. It doesn't work. I tried that. And actually, I had a student that went to Denmark for 3 months trying to kind of standardize one protocol for pipeline, to develop a pipeline for nanopore assembly. It didn't work. So let's ditch that. So I introduced 7 years ago the sequel 1, and it stayed 7 years in my lab, and we published more than 60 publications during these 7 years. Many genomes have been deposited in the NCBI database. I'm going to talk about one particular project because I needed to sell my kidney to pay for the sequencing on sequel at that time. So Candida auris is a yeast that is very popular now in the States. This yeast also had a kind of an outbreak back home in Lebanon. And then I collected 29 samples from this outbreak. At that time, there was only 80 Candida auris genome in the database. This includes complete and draft genomes as well. And now there is almost 450, and 50 of them are complete genomes, out of which we deposit 30 of them. So more than 50% of the complete genomes in the database was done on the sequel, just to produce this really nice phylogeny. So the red ones, as you can see, these are our samples that we put, and you can see it. It's a 7-chromosome diploid organism, and we managed to separate the different clades, which we have 5 clades in this, and then it kind of separated real nice. The variation between the isolates in this clade is up to 5 SNPs per genome. So with the coverage that we had, so we were sequencing 2 samples per run in the sequel, so I had 15 runs. And now we have more than 50% of the database sequenced by PacBio. But then they convinced me to buy the Vega and the Reveo, so we ditched the sequel one, and now we have the Vega and the University, and I started my company Genomics, which is a core facility sequencing long reads using Reveo. I'm going to talk about one project which is not published, so I don't have all the data in here. But I wanted to talk about Acinetobacter baumannii. Maybe I can convince you that microbiology is really important and you should invest more in the R&D. Right? Yes. So Acinetobacter baumannii is a gram-negative bacteria that is firstly known for its being very aggressive, and it's actually the first bacteria to have the superbug characteristics. So there exist Acinetobacter baumannii that are resistant to all available antibiotics. This started to be popular during the Iraqi war because you can get it from the environment, and normally soldiers who have been injured, they got back with a lot of infection of Acinetobacter baumannii. This is an outbreak back in Lebanon in the ICU unit, and they had to kind of isolate part of the ICU, and any inpatient walking onto the emergency that is screened for Acinetobacter, and it's positive, he will be isolated there. The mortality rate in the ICU in that section is 96%. It's very aggressive, and you will get fever in 1 day, and after 24 hours, you will be taking antibiotics, but it doesn't work, and you will die. So the hospital know that I work with microbial genomics, so they called me and they told me that they want to know what is the hidden source of this Acinetobacter baumannii in the ICU, because they're doing, environmental swabs from the hospital and disinfection, but nothing is working. So I did some environmental swabs. As you can see, I had some positive swabs, and this was after disinfection. So it's side rails of the bed in the ICU, air mattress, the monitor, and also from the toilet. All belong to the same sequence type. And these were extreme drug-resistant antibiotic strains. Only it worked with colistin. But even with patients taking colistin, it was not working because you're an ICU patient, so you already don't have a good health, and then the septic shock kicks in, and then the antibiotic does not work anymore. So I made some really nice sequencing, as you can see, comparison. Everybody was, especially Nanopore, wanted me to buy the Promethean, so they gave me free cells to sequence my isolates. So I did some comparison. I don't have much of the information because this was sequenced over the weekend, so I'm just going to talk a little bit about the problem that you have in Nanopore. And normally this is in the assembly. So you could see that doing the assembly using fly, and the Promethean coverage is 600x. So I used fly to do the assembly, and then I did a unicycler to do a hybrid assembly. As you can see there, that there's a lot of number of bases that are lost when you do the hybrid assembly. And another problem that you will see is that with Promethean, you will always lose what we call the small plasmids that is really important for the virulence. And this is something that I noticed on hand. And this is why when I did the run on the Reveo, I used I don't use any size selection with the library prep, because there are some plasmids that are 1.5 kb or 2 kb that I don't want to lose. And also with Promethean, you don't have any size selection anyway, but when you do the assembly, you will lose all these very short reads. And you can see that you cannot see it in the Promethean. Some of them is, again, discovered when you do the hybrid assembly between Illumina and Promethean. Another fun fact that we did, I did what we call SNPy, so you have a reference genome, as you can see upstairs. And then I did mapping of the reads from Illumina alone, from Promethean alone, and then from the hybrid assembly as well. You can see the SNPs is 0. You had 2 SNPs of Illumina and 1 SNP of Promethean, but when you do the hybrid assembly, that goes away. So you say it's really good, but the assembler and the extra polishing does not always work. So you had one snip with this sample and one snip with this sample, and then when you combine, you have 4 snips. And this is because of the extra polishing. Normally, this is what goes wrong. But again, deletions is also always solved. And just for the fun of it, I wanted to compare the HiFi used on Reveo to compare it with the sequel. And it seems, even though it's the same DNA, it's 2 different bacteria, as you can see with the high number of SNPs, insertions and complex insertions. And also, you would see a lot of different gaps if you compare the sequel reads with the long reads from the HiFi. So just to sum up now, with the introduction of Vega, I think everybody working in the microbial genomics should ditch the short reads. Now the price per sample is really nice. Maybe if we're gonna talk a little bit what we can do better. HiFi reads, from my perspective, it's still the best reads that you can produce, even when you compare it with Illumina. And the most problems that you can get from other platforms for long reads is that you don't have a standardized approach in how to do the assembly, and normally the assembly breaks. And what we'd like to see in the microbial genomics, and this is for you, Neil, so we need the RNA sequencing for microbes. The experimental one does not work. We should update the microbial assembly. It works really nice, but I think with the advancement in the technology, maybe we should do some upgrade. For Vega, I know that you wanted to show that you're going to kind of expand the smart cell. I would like to see a smaller smart cell. Yes, I mean, you know, I could wait for the whole year to collect 380 samples, but that will cause a bit of delay, so maybe, I don't know. Of course, keeping the same price. No, I mean the price per genome should be the same, let's say. For the sequel, this is what I used for this HiFi reads. It's really good, but there should be kind of protocol for fragments above 10 kb without the size selection, because the mean was almost 8 kb. So sometimes if you don't have enough coverage, you will have a bit of broken assemblies. And I would say reusable smart cell, but that's already on the way. So thank you for having me, for my colleagues, Dominic from the distributors of PacBio and Czech Republic, and for your time and your patience with me and the microbial assembly. So thank you.